Hey, good evening, everyone. Very nice to have you here. My name's Matthew Scott. I'm president of the Carnegie Institution for Science. And welcome to the very first Carnegie Evening Lecture of the year and the first one in this newly renovated auditorium. We have had a team of craftspeople of many kinds working together very closely for months to change almost everything about this auditorium except its historic aspects, like these wonderful murals and the phases of the moon and the sun overhead. And this is the first big event here. It would be somewhat bigger in terms of the number of people if the entire city wasn't in gridlock right now. So congratulations to all of you who made it. We have 550 people signed up, and that's far more than have been able to reach here, but we didn't want to keep you waiting anymore. So we have doubled the number of these lectures this year to 10 from 5, and we hope you find many of them very interesting, and we hope you'll join us for all of them. Now, clearly, there's huge interest in the science of tomatoes and tomato chemistry because we had 500 people sign up in advance and had to turn people away. So it's great. It's gratifying in general for us with these lectures to see such community interest in science. It lifts our hearts, we who are practicing scientists. I do recommend that you check out our renewed new website, which I think gives you better access than the old one to many of the scientific projects we do. It's been a fantastically exciting past year with our airborne observatory flying and our telescopes hard at work in Chile. It's just everywhere you look, all sorts of things are happening. And we have, in the previous slide, which disappeared, but you were looking at it before, a new Carnegie Science logo that celebrates our connections between space, earth, and life, our sort of three realms of endeavor for our institution. So I'm very happy to be able to do an introduction about the life part of that tonight, which is what we'll be hearing about tonight. And we can go to the next slide and see what we're talking about, sort of an attitude of our views of the integration of the sciences, as it were. So more than half of the vegetables consumed in America today come from just two sources. Now you're thinking about which two: potato and tomato. Chips and French fries, not necessarily what we would consider vegetables, predominate, but tomato sauces are a close second. Salsa and ketchup battle for the top spot in supermarket sales, while in prepared food eaten outside the home, the red sauce and pizza wins handily. Despite the tomato's popularity, however, the disappointment one experiences in tasting a supermarket tomato is nearly universal. The tomato has a remarkable and roundabout history. Scientists believe it is native to South and Central America, but came to North America only after passing through Europe, to which it was introduced by Spanish conquistadors in the 1500s. People living in Mediterranean climates found that the tomato grew well in their gardens, and it quickly became a culinary staple. It took people living in other places, particularly in England, some 200 years to shake the mistaken perception that the fruit was poisonous. This is a remarkably slow learning process, it seems to me, but I mean, poisonous things get eaten faster than that, but anyway, I'm supposed to stick to the script here. In 1728, a professor of botany at Cambridge University declared that the tomato makes, quote, an agreeable plant to look at, but the fruit of most of them is dangerous, unquote. I think that's more true now in supermarkets than anywhere else. This attitude made its way across the Atlantic, where Americans were slow to recognize the tomato's virtues. Many people, including Thomas Jefferson, grew the plant, but only in their flower gardens. Others found purported relief from various ailments by ingesting tomato extract pills, which were widely advertised as a universal panacea. By the mid-19th century, tomatoes were being eaten in America with gusto, with recipes for stuffed, hashed, pickled, baked, broiled, and preserved tomatoes widely shared. Ketchup had been invented and successfully marketed, foreshadowing my son's staggering capacity for disposing of it. He's a rugby player, and you just would not believe. By the end of the 19th century, the tomato had the unique distinction of being the subject of a Supreme Court case. In Nix versus Hedden, the justices decided that the tomato, though botanically a fruit, should instead be categorized as a vegetable. And so it has remained, but for the cognoscenti, that's you. But if a tomato is a fruit, is ketchup a jam? A deep philosophical point I thought I'd throw in there. Today, thousands of tomato cultivars exist worldwide, with the majority grown for the commercial market. In 2012, tomato production was the 8th most valuable agricultural product worldwide, with an industry valued at nearly $60 billion. That's an amazing number, isn't it? That's for one kind of plant. Of course, it's not one kind, it's many kinds. The big tomato growers favor tomatoes with consistent size, shape, and color, disease resistance, a firmness suitable for mechanized picking and shipping, and long shelf life. The tomatoes have been bred to have many of the salutary properties of cardboard. What is often missing in these tomatoes is what our guest tonight has studied for over 20 years: flavor. In his laboratory at the University of Florida, Dr. Harry Klee and his colleagues, including a psychologist, have identified many of the volatile chemicals contributing to flavor in dozens of tomato cultivars, including heirloom and supermarket varieties. By enhancing the concentration of some of these chemicals through traditional breeding methods, i.e. mutation and selection, Dr. Klee's group is creating new tomato varieties that have the rich flavors of homegrown heirloom tomatoes, combined with the sturdiness and consistency required for commercial success. As a bonus, when commercially processed, these flavorful tomatoes don't need added sugar, thus reducing their caloric content. A member of the National Academy of Sciences, Dr. Klee is the Lyle C. Dickman Eminent Scholar Chair in Plant Improvement at the University of Florida. I would regard it as greater than that, not just plant improvement, but human and life improvement was such an important thing. He's a faculty member in the university's Horticulture Science Department. He studied psychology as an undergraduate at the University of Massachusetts in Amherst and went on to earn his PhD there in biochemistry. He worked at Monsanto for 10 years and then moved to the University of Florida, where he explored the molecular biology of ethylene, which is the chemical that causes fruit to ripen, before transitioning to studies of flavor. He has studied flavor not only in tomatoes, but in melons, strawberries, and lettuce. Those will be 3 future Carnegie evening lectures, with adequate sampling as well. Flavor, he has said, has proven to be a really hard problem. It's fascinating when someone undertakes something we're all sort of familiar with and has been tremendously understudied, and they start to reveal all the wonders and intricacies of it. And that's what's happened here. It's proving hard to sell flavor to the big tomato growers, so you can all write letters after this talk. Already, Dr. Klee and his colleagues have produced at least one new hybrid tomato variety that has all the qualities attractive to industry and tastes good, but for now, the big tomato companies and supermarkets are not paying adequate attention. As consumers, we can only wait and be confident that tomato greatness is just around the corner. Please join me in welcoming the person who we have no doubt will be largely responsible, Dr. Harry Klee.
Okay, is this on now? Okay. That was probably the best introduction I think I've ever had. Oops. Yeah. Okay, so how do I get, there it is, okay. It's a real honor. Scientists have one of the biggest challenges scientists have is to talk about their work to lay audiences. It's an extreme challenge, and I have to say I've been more nervous about this talk than I was about probably anyone I've given in 10 years. So let me just dive into it. Yeah, I think this is probably there. So we work on tomato flavor. Obviously, the crowd that's turned out says how important this is to people. When you think about flavor, my daughter, who's actually in the audience, says, do people really care about flavor? Well, I think that this audience is probably proof that, yes, they do care. It is a hard problem. It's taken us a very long time to figure it out. And I don't want to go into all the details of a lot of the science. I'll try and give you a flavor for the approaches we're using, but I really want to tell you the whole story at a sort of 10,000 foot level. So what's the problem? Let's set up the problem here. The number one complaint in the supermarket is tomato flavor. Everybody says they're lousy. Why are they lousy? Everybody is uniformly in agreement on that. So what's the problem? If you think about it, let's start with the breeders. The breeders are the guys who are developing the varieties that you are buying in the store. The customer of the breeder is not you. The customer of the breeder is the grower. And the grower, for the most part, is not paid to produce flavor. The grower is paid to produce 25-pound boxes of round red objects that can be shipped great distances. And that is what the grower cares about. We've had conversations with the growers, and the growers say, well, yeah, well, first of all, they say, oh, there's nothing wrong with my tomatoes. And I say, well, we've got empirical tests that say that's not true. And they say, well, fine, but you've got to give us good flavor in the context of something where yield is king. I have to have yield because yield pays the bills. So you can't fault the farmer, and you can't fault the breeder for this. They're doing what they have to do to make money. So they're paid for yield, not for flavor quality. Now, one thing I put up here in red that I want to illustrate, I won't really go into this tonight, but tomato is kind of like the canary in the coal mine in one sense. A lot of modern agriculture has focused on yield, and there are some really interesting studies out there that say when you focus on plants, developing plants that just produce more and more and more product, what you do is there is a trade-off with nutritional quality. And tomato is a great example. Strawberry is another example, where when we produce more and more product, what we're basically doing is adding water. And so what you end up with is products that have lower, on a weight basis, lower nutritional quality. So it's not just tomato. The problem is this, so this is what's driving the people. It's yield, it's shelf life, and this is how many of you refrigerate your tomatoes? Come on, admit it. You shouldn't do that. It ruins the flavor. It destroys the flavor. But I can't tell you how many times we've gone to supermarkets to get control tomatoes for our consumer panels, and the produce guy goes out back and he brings out a box and it is ice cold. They have one temperature that they can store things at, and it's 4 degrees Celsius. And so they put their tomatoes in the refrigerator. Again, because they think they can do it, because they're more focused on keeping that stuff alive and looking good than on tasting good. So that's really the heart of the problem. But you are not blameless in this. What you want, despite what Alice Waters will tell you, what you want is you want cheap and you want it year-round. You don't want to eat tomatoes in August and September and then not again until the following August. So the system has been set up to give you what you want year-round. And people, you can get a decent tomato today in the supermarket if you're willing to pay for it. Most people are not. So it costs money to produce something that tastes good. Our real challenge is to produce something that isn't going to cost any more but tastes great. So that's our gold standard. That's where we want to be in the end. But the reality is, if you go to your supermarket and say, I'm not going to buy this junk, and I'm willing to pay a little more for something that tastes good, I promise you they will try and get it. Now, they may have difficulty finding it, but they will try and get it. So that's the number one problem. The number two problem is if you think about breeding for flavor, what is the assay for flavor? You have to put it in your mouth and you have to chew it. If you've ever grown a tomato in your yard and it's rained and you've gone out and picked that tomato after it's rained, it doesn't taste as good. It's got diluted out flavor. The same plant on two separate days can taste very, very different. So and as you'll see soon, it's a complex genetic problem. It's many genes that control the chemicals that contribute to flavor. So we have a problem in that it's a very large number of genes controlling this trait, and the assay is very difficult. Again, the assay, think about a breeder who's got 100 varieties out in their field, and they're going to go out and they're going to pick 100 tomatoes in the morning and taste them all. You can't do it. So what has happened is the breeders have basically thrown up their hands and say, it's too hard, I can't do it. So what they've done is they have neglected flavor. And when you neglect any trait, any complex trait like this, over time it deteriorates. And that is exactly what's happened with the modern tomatoes. So our real challenge is to make a tomato that tastes really good but maintains the commercial properties that they're going to insist on having: yield, disease resistance, firmness. So we need to make that good flavor without significantly increasing the cost of the product. And that is a big genetic challenge. So I talk about nutrition a lot. And the reality is people eat what they like. Why do people eat at McDonald's? Because it's cheap and they like the taste. What has happened to modern foods, modern produce, in many cases, is that the flavor has deteriorated. How do you get people to eat a better diet? You make the foods that are good for you taste better, rather than having an industry that's putting chemicals into your food to make you think it tastes better. We have to make these natural foods taste better, and if we do, I think people will make more intelligent choices. We've talked a lot about this with people in the medical school and in extension. We're actually really excited about potentially doing some in-home studies. I think the reality is if we give people better tasting produce, they will eat more. Anecdotally, we know it's true. We'd really like to prove that. So we've taken a rather unique approach to breeding in our program, in the broader program at the University of Florida. And what we are trying to do is we're trying to say, look, the problem with most of these modern foods is the consumer has never been asked. The breeders don't go out and ask the consumer, what do you want? One of my colleagues is very fond of saying that in ornamentals and flowers, 90% of the flowers are bought by women. 90% of the flower breeders are men. There's a disconnect here. And I think the tomato, again, is a really good example where the breeder is completely cut off from the consumer. So we're turning that around. And this is where I collaborate with a really great psychologist, Linda Bartoshuk, who's one of the world's experts on flavor and taste and measuring people's responses to things like foods, and particularly in smell. And so what we're trying to do is we're trying to institute a system where we're asking people, what do you like? And I sort of capture that on the next slide. We start with the consumer. What is it that the consumer wants? And then we do it by rigorous sensory analysis. We use, in our case, consumer panels of 100 people. And we take measurements of what people like. We take a lot of measurements about who they are, some of their genetics in some cases. But what do you like? And then we work backwards. And you'll see as this process unravels. We go from what do you like to what's in what you like. And then we try and draw the correlations with what are the chemicals that drive liking. What are the chemicals that make you like this tomato versus that other tomato? And then we work back from there. How does the plant make those important chemicals? What is the genetics that controls that process, those biochemical processes? And then ultimately we get to the breeder, and then we go all the way back again. So it's a cycle where we're saying, what do you like? What drives what you like? What's the genetics behind that? Let's turn that into molecular markers for the breeders, and then let's go back and retest what we make with the consumers. So that's the whole process. So what is tomato flavor? It's actually a mixture of a number of different chemicals. Now, if you think about something like banana, it's a very easy product to work on. There's one or two chemicals that really stand out. I could give you a little vial of one specific volatile. You'd smell it and you'd say, it's a banana. It's very difficult to do that with a tomato because there are a lot of different chemicals that all interact to give you that overall palette that your brain says this is a tomato. We know that sugars are really critical. People respond to sweetness. And the reality is, in many cases, the sweeter the better. So glucose and fructose are really critical to flavor. We know that there have to be acids. The sugar to acid balance is very important, and different people have slightly different preferences for that sugar to acid balance, but overall, a tomato that doesn't have any acids is going to be very bland. But where we really focus most of our effort is on volatile chemicals. So the volatiles, in the case of the tomato, if I use a gas chromatograph, I can tell you that I can see 400 different compounds. Sorry, wait a minute. Yeah, kind of fades out here. About 400 different volatile chemicals I can detect. So that gives you an idea of the magnitude of the problem. Well, it turns out it's not quite that difficult. What we've gone through is we've gone through and sorted out all of the chemicals that we can detect and say which subset of them really drive consumer preferences. So if we think about smell, there are only 5 classes of taste receptors in your mouth. There are only 5 different kinds of chemicals that you can taste in your mouth. Well, think about the foods that you've eaten just today. There's a whole lot more than 5 flavors that you've experienced today. Virtually all of flavor really comes from smell. So it comes from olfaction. So what happens is we put something in our mouth and we chew it. Oh, well. Oh, yeah, there it is. So we chew something in our mouth. We are detecting sugars. We are detecting acids. We're detecting sour, sweet, salty, bitter. But what happens is that when you start to swallow, air is forced up through the back of your mouth to your olfactory epithelium, where a whole set of about 350 different receptors are there to detect the compounds that are the volatiles, the things that we smell. Think about the last time you've had a cold and how you couldn't really taste anything. That's because you couldn't smell. If the olfactory system is blocked up, you pretty much wipe out. People who've had neurodegenerative problems that cut off the olfactory system cannot taste anything. It's actually a very common problem. So smell is really what we care about. And smell is really what drives our research, and you'll see why in a few minutes. So I'm going to put a list up here, and there are a bunch of chemical names, and I don't expect you to remember any of them, but I want to make a couple of points. This is a set of very diverse chemicals, but what I want to tell you is this was something, when we set out to discover what makes a tomato taste good, we actually have discovered some really interesting basic principles about taste and human responses to foods. One of the things we discovered is that virtually all of the important tomato volatiles, so I'm skipping ahead and showing you many of these important volatiles here, virtually all of them are derived from compounds that are essential to the human diet. Essential amino acids, essential fatty acids, and carotenoids. The fact is, what this suggests, and this is a very difficult thing to prove, but we believe that what we like is because it's a signal that it's good for us. So all of these chemicals, all of the major volatiles are derived from chemicals that are directly related to things we need in our diet. We like what is good for us. So that was one of the early discoveries that we made about this whole process. But anyway, a very diverse set of chemicals. So that suggests that it's going to be a very difficult problem in the long term. So how do we address that problem? The way we've done it is, and I'm not going to go into a lot of the quantitative genetics, but we've taken advantage of the resources that are out there. So again, this was a great introduction. All of these varieties that we work with are wild species, wild relatives of tomato through South and Central America. And they're very, very different from the domesticated tomato. There's a lot of genetic diversity and there's a lot of chemical diversity. So, and the nice thing is we can make genetic crosses with virtually all of these species and get fertile progeny. So we can actually take apart the tomato genome piece by piece by using the relatives to understand the regulation of these important pathways. But where we really made a surprising breakthrough to us, now, again, in the introduction, you got a great hint of this. Tomato has gone through a couple of different domestication bottlenecks. One by the natives of Central and South America, and a second by the Europeans. Despite that, there is a tremendous variation within the tomato population. These are all different heirloom varieties that we grew in one particular season. And I will tell you that the variety that you see in terms of shape and in terms of color is also reflected in flavor. So we have this tremendous variation within the tomato, within the species itself. So within the genus and the species, we have this tremendous genetic variation and chemical variation, and we exploit that variation. We want to understand what people like. We want to get the range of things from very good to very bad. And I can promise you there are a whole lot of heirloom tomatoes out there that really are quite bad. And, you know, someone says, what's the definition of an heirloom? Well, there actually is no definition of an heirloom. There is no legal definition. Heirloom basically means it's an old variety that people threw away because they found something better. So we've gone back and we've exploited those heirlooms. And I want to show you, this is just a schematic of think of this, the vertical bar as our taste scores. So we give these tomatoes to people and they give a score, and up the very top is the best tasting tomato ever, and at the bottom is the worst taste. And what I want to show you is that a couple of points. First of all, small tomatoes are better than big tomatoes. And so if you go to the supermarket and you really want something that tastes good, get a cherry tomato. Don't buy those big monster tomatoes. They have more flavor. Consistently, we see the smaller tomatoes have more flavor than the larger tomatoes. So the very best tomato up there, Cherry Roma, that's pretty much the best heirloom we've ever grown in terms of ratings. But down at the bottom is something called Matt's Wild Cherry, and I see people growing this all the time. You see it in magazines. People say, oh yeah, I grow this. It's a tiny little thing, and they're terrible. In fact, they're disgusting. The best big tomato we've ever put through our consumer panels is something called a Brandywine. I'm sure many of you have heard of Brandywines and probably even grown them. But down at the bottom, I did a sabbatical in France, and they said, oh yeah, in France, of course, everything's better here. And we grow great tomatoes. Well, Marmande is a French heirloom variety. Well, it's the worst tomato we've ever put in our consumer panels. The point being that there's a huge range. That works to our advantage because we can exploit that range to understand human responses and to understand genetics. So I just want to show you a few of those chemicals that were on one of those earlier slides, and I want to make a point. Between different tomato varieties, we see huge variation. Now, some of this is environmental, but much of it is genetic. So we're seeing thousands to hundreds to tens of fold difference. Well, what does that mean? It means that the genetic diversity is out there. We just have to understand it, and we have to put the puzzle back together with the right combination. But the genetic potential is there if we can just identify it. So the good news is most of the modern varieties, the commercial tomatoes that you're buying in for 99 cents a pound in your local supermarket, are at the lower end for most of the chemicals, with a couple of exceptions. But for the most part, we again have just basically diluted out flavor as we've grown varieties that produce more and more fruit. Basically what we've done is we've produced plants that are producing so much fruit that the photosynthetic capacity of the plant has been exceeded. And it just can't fill those fruit with sugars and acids and volatiles fast enough. So we know we know that the genetic diversity again is out there, and we know that the commercial tomatoes are at the bottom end. So we know it's an easy target. So I want to tell you, I realize there are many people, I know some of your scientists are going, oh my God. I need to introduce one term to you, and it's alleles. And alleles are extremely important. So all of you know about eye color. Eye color is the classic genetic, we all have different alleles of genes that give us different outcomes. Well, tomato is exactly the same. All of these are different varieties of tomatoes. Just the way different alleles of a gene give you different eye color, different alleles give you different fruit shape, different fruit color, and different chemical compositions. So our goal is to, number one, understand which genes are important, and then number two, to identify the alleles that give us the desired property. Whether you want the blue eye or the green eye or the brown eye, we have to do that. We have to identify the right alleles of all of the important genes. So that, in a nutshell, is the process. Identify the chemicals, identify the genes that are responsible for making them, and identify the alleles that give us the good flavor, not the bad flavor. Okay, so this is what we're trying to do. We want to take flavor down to the level of a DNA test. Just the way you send your little mouth swab off to 23andMe and get your genome sequence done, we want to be able to do that with the tomato. We want to be able to define a set of genes that are important for flavor and then identify the best alleles and be able to translate that into something that the breeders can work with. So they don't have to go through and taste their 100 or 200 different varieties and say, this is good, this is bad. We will be able to reduce it to a DNA test that says this one is going to taste good, this one is not. So that is the ultimate goal. So how do we do that? The first step is, what is flavor? What are the important chemicals? So here, we have a great collaboration with Linda Bartoshuk, who's shown up there, and Charlie Sims, who runs the food science lab at UF. And what we basically have done is, we had been working on the genetics for a number of years, and then all of a sudden, it just kind of dawned on us, we've got this incredible, diverse resource. We need to define the targets. So we took some of the heirloom tomatoes that had the greatest range in flavor, from very good to very bad. We have a 100-person consumer panel that comes back throughout the season and tastes these tomatoes, and they rate them. And they say, this is a great tomato, this is a good tomato, this is a bad tomato. Well, at the same time, in our lab, we're grinding a subset of those same tomatoes up, and we're saying, these are the sugars, these are the acids, and these are the volatiles that are in those tomatoes. It becomes a statistical problem. We basically are extracting out statistically what is in the good ones, what's in the bad ones. And we can do that, we've done it, and we can develop a statistical model that defines the chemistry of liking, basically. We can develop a recipe. If you have this level of this chemical and this level of this other chemical, and we put them all in combination the right way, we will get a tomato that tastes great. So we've done that. We know exactly what the target is, and we have a recipe. So just to summarize the results, first of all, I always get asked this question, so I'll answer it preemptively. There are differences. We have 100 people. It's a fairly diverse group. We can't break all of it down statistically in terms of different ethnic groups, for example, but we know that there are ethnic differences. We know that there are age differences. Older people like a different tomato than younger people. We know that women like a slightly different tomato than men. We know that foodies like a different tomato. So we ask people lots of questions about themselves. We can tell who are the food lovers. The way one of my colleagues says, there are people who live to eat and people who eat to live. And we can separate those people out, and it turns out they like different tomatoes. That said, the average of 100 people is a great place for us to be. It gives us a recipe for what everybody thinks is a very good tomato. It may not be your favorite, but it'll be up there. So that we've done that, we've defined it. We've defined a set of about 26 different volatile chemicals plus sugars that really positively and some of them negatively contribute to flavor, to liking. Sweetness is really important. And this again, so here's another case where this was an unexpected discovery. And this is the great part about doing interdisciplinary science, working with a psychologist. We knew that sweetness was a major driver of liking. I mean, there's a reason that Coca-Cola makes a whole lot of money. People like sugar. And so what was surprising to us, though, was we discovered that specific volatile chemicals. Now remember, again, that if you know anything about neuroscience, this is pretty amazing because taste occurs in your mouth, smell occurs in your olfactory epithelium. Volatile chemicals are enhancing the perception of sweetness. So this is occurring at the level of your brain. So your brain is pairing specific volatile chemicals that we find in tomato, and we've also done this with strawberries, we've done it with blueberries. There are specific chemicals that enhance your ability to perceive sweetness. Well, that has some really interesting implications, and I'm just going to show you one slide here. So this is a correlation of a number of different heirlooms that we've tried, comparing perceived sweetness. So that means your rating of how sweet you thought that tomato was versus the actual levels of sugar. And you see the two tomatoes I've circled here. The yellow jelly bean actually has more sugar than the matina. Yet people are saying that the matina is over twice as sweet as the yellow jelly bean. That's all volatiles. And we can show that the ones that are above the line are enriched in specific volatiles, and the ones below the line are depleted in those volatiles. Where this is really important is it means that if you're in the food industry, you could potentially reduce the amount of sugar, replace it in certain foods with some of these volatile chemicals, and you could reduce the caloric intake dramatically while maintaining the same level of sweetness. So that's really interesting. But again, it tells us these particular volatiles are really important, and they're important because they enhance the perception of sweetness. Okay, so just to summarize that part, we know which volatiles make important contributions to liking. We have the recipe for a great tasting tomato. So we've defined the target. So we quantitatively and qualitatively know where we want to be to make a great tomato. Now the question is, how do we get there? So how do we get to the important genes? We use a combination of biochemistry and genetics. And I'm not going to talk to you about the biochemistry, but I'm going to show you one slide, and I promise don't freak out. Everything in red is one of those volatiles that we work on. And the point being that we've put a lot of work into understanding how the plant makes those chemicals. Why is that important? It's important because we need to understand the pathways, and if we understand the pathways, we can potentially go to what are rate-limiting steps in those pathways. So, for example, that pathway over on the the bottom left, if we understand what the rate-limiting step in that pathway is, we could potentially coordinately affect 7 or 8 different volatiles. So by understanding how the plant makes these chemicals, we can potentially leverage specific points within the metabolic pathways where we can get more bang for our buck. So why that's important? Here's the list of the 26 volatiles that we can show are really important for tomato flavor. You say, oh my God, I can't work on 26 genes. The breeders would say, forget it, I'm not even going to try. Well, by knowing a little bit about the biochemistry, knowing about the coordinated regulation, we can actually reduce the problem. So we can reduce the problem because within one of those circles, we can get a single gene that impacts all of the ones within that circle. So suddenly, we can get almost all of these volatiles with only 5 different genes if we're careful and pick the right gene. So that reduces the problem dramatically in terms of a breeding strategy. So how do we identify them? Well, we've leveraged what in the last few years has been a remarkable revolution in biology, and that is cheap genome sequencing. It's really changed everything. We've spent 10 years doing genetics one gene at a time the hard way. Well, it turns out that with cheap genome sequences and a partner in China who doesn't charge us anything for genome sequencing, we've been able to get the genome sequences of about 350 different tomato varieties. So we have complete genome sequences of all of the heirlooms that we've worked on for all of these years and a whole lot more. And it hasn't cost us a penny. It pays to have collaborators in China because they have way more money than we do. So what we do, we get the complete genome sequences of all of these varieties, the ones we put through the consumer panels. We quantify all of the chemicals, and this is actually the hard part. So in the last few years, just in the last 2 years really, the equation has totally flipped. It used to be that genotyping was limiting to everything we did in genetics. Now it's the phenotyping. The phenotyping is what's limiting. We are literally, believe me, doing all of these chemical analyses on any one variety takes a lot of work. We can maybe do 50 in a season. So for us to do 350 is a major effort. It's totally flipped. Now these guys have got the genome sequences before we've barely even harvested the tomatoes from the field. So that's changed dramatically. So quantifying all of those flavor chemicals really is what's the holdup right now. And then what we do is we look for correlations. So just like with the consumer panels, you've got a genotype, you've got a phenotype. You look at the alleles of the important genes, and they just pop out. So I want to show you just an example of this, 2 examples actually. What you're looking at here is a representation of the 12 tomato chromosomes for two different traits, one volatile and in this case an acid. And that red dashed line that goes across there is what's highly significant. If it's above that line, it's highly significant. So you can see that for citric acid, for example, right here on chromosome 6, at the bottom of chromosome 6, there is this genetic locus that is hugely correlated with the content of, in this case, citric acid. In this case, we've got one that's highly correlated with this volatile chemical guaiacol. So you literally do this for every chemical that we're looking for. Now, it's a work in progress. We've identified a number. So we don't get the exact gene in this case. What we've got is a region on the chromosome that if we have this region right here, if we have the right allele, so what we're looking at is the computer is literally going through and saying, I've got two alleles, and I'm going to go and I'm going to look at every allelic combination throughout the entire genome, the 12 chromosomes, 30,000 genes, and I'm going to look and see, is one allele correlated with high citrate and another with low citrate? So it's going through literally every single nucleotide change on every single one of these genotypes and looking for these correlations. So it's been extremely powerful for us. Literally, this is just a screen capture where we go through, we can go down to the nucleotide level of any gene, and we can display what's different about those genes in any given genotype that we type in. So literally, it's now become a computational problem. It's really now the rate limiting step for us is bioinformatics. So I just want to show you a few examples. What you're looking at are specific two different alleles that we identify using that process. So for example, soluble solids, which is a proxy for sugar content. You can see one allele, two alleles. So you can see we're getting about a 10% increase if we have this allele. Citric acid, we're getting a little bit more, I don't know, maybe 20, 25%. So you go through here, there's a very big difference if you have one allele versus the other. We are literally, so this is where we are today. We are literally going through the entire genome and we're saying for chemicals A, B, C, D, which allele will give us the best concentrations. Now, because much as I would love to do this by GMO approach, the reality is we can't afford to do it, and we're not going to go through that. So we are using, I like to tell people, and this is a really good example, if I could use GMO and I could afford to use GMO, you'd have your great tasting tomato today. Because we are not going to do that, we are using traditional breeding. It requires several years. So that's where we are today. We've identified all the right alleles for most of the traits that we're interested in, not all of them, but most. Now we have to go and we have to use genetic crosses to combine all of those things. And you can imagine, we're talking now 6, 7, 8 different genes. To get them into a single genetic background is complicated. It takes a lot of time. It's mostly time. So it's going to take us a couple of years before we can stack up all of these genes in a single variety to really improve flavor. But we know how to do it. We know what the pieces are. So we've gone through systematically and said, this is what we want to target. This is how we're going to target it. These are the tools that we're going to use. Now we're in the process of using those tools. Okay, so it turns out that there are a few low-hanging fruit, and as I kind of hinted to you at the very beginning, the hardest problem of all is the commercial tomato, that bulk tomato in the big bin that you're going to walk into your supermarket and see. Because we have to get all of these chemicals back up to the right levels without compromising any of the agronomic properties of those plants. Well, it turns out there are some easier targets for us. And people like you are those targets. Home gardeners. again, we had one of those Homer Simpson moments a couple of years ago where we said, we know a whole lot about heirlooms, and heirlooms are really, really hard to grow. They're susceptible to diseases, they're soft, their yields are crap. Can we fix those tomatoes? And the answer is yes. So we decided that we would set up a program in parallel to that commercial target of giving home gardeners tomatoes. So the dirty little secret that the seed companies won't tell you, all those varieties, not the heirlooms, but all of those things like Big Boy and Better Boy that you might have grown in your yard, those are cast-offs. They're the ones that weren't quite good enough for the commercial products. And someone said, I bet we could sell those to home gardeners. Well, again, we're focused totally on the consumer. We've learned a lot about which are the good varieties and which are the not so good varieties. So we said, can we leverage that information to develop varieties that really, really taste good and have much better performance? So what we're trying to do is, can I capture heirloom flavor and modern cultivar performance? And whether it's 90% of a commercial hybrid doesn't matter to you. What you want is lots of good flavored tomatoes. More, hopefully, than you're getting off those heirlooms. So we've done that. So I just want to show you this. This is, again, that slide of liking. The first variety that we've developed we call Garden Gem. And it's heirloom parent. There's the modern parent. So it's a hybrid. It's a hybrid between a modern, disease-resistant, high-yielding, high-performing variety and a really good-tasting heirloom. We got all of the flavor of the heirloom, and we get in fact, we get just as much yield as we do off the modern parent. So we really did, in a few cases, and I will tell you, we did a lot of crosses with a lot of heirlooms and a lot of parents, but we've got a few that really perform. So on the left, you're looking at the heirloom parent, and in the same field, you're looking at the hybrid. The tomatoes that come out of that are significantly larger. This was actually a cherry variety. It turns out that the hybrid is about the size of a Roma tomato, the ones you'd use to make tomato sauce. So we've got tremendous yield and all of the flavor of that heirloom. So we call the first one Garden Gem. It's about a 50-gram tomato. It's got great flavor. We know that people want large tomatoes, so we're also doing that too. The other one, the Garden Treasure, is a large one. It's a 200-gram tomato. Again, modern performance, but heirloom flavor. So the growers just love to say people don't care about flavor. So what we did was, I actually visited Rutgers about a year or two ago, and they said, we always get asked for our varieties, that people can't get those Rutgers tomatoes that were developed back in the 40s and 50s. They're off the market. So we decided that we would start to produce them and sell them to consumers. So we said, that's a great idea. Until a seed company really picks these things up, let's just put them out there. So word of mouth, no advertising really. Since March, when we put these up on the website, for just a small donation, we send these seeds as a gift. 1,600 people have made contributions. We've hit 48 states. We've hit 22 countries. There's just this incredible backlog of people who are dying to get their hands on tomatoes that taste good. This just tells you that, I just don't know how to say it any simpler. The growers are just wrong. The supermarkets are missing a huge opportunity here. And the growers are just blind to flavor, and this has to change. And it has to really start with you. It has to start with these 1,600 home gardeners who are saying, I'm going to grow my own great tomatoes. I can tell you a lot of these people have heard about them. They say, I've never even grown a tomato from seed. I go to Home Depot and buy whatever they've got. But I'm going to try yours. And I end up spending a lot of my time telling people how to start tomato seeds. But it's a labor of love, so it's okay. And just in case you were wondering, North Dakota and South Dakota. Apparently, they don't like tomatoes. So, just to conclude, I think that we've made really good progress. Now, I'll tell you, this is the reason that I left Monsanto. I left Monsanto 20 years ago. A company would never do this kind of research. It's too long term. It's been a real challenge, but I really honestly feel like what you're hearing tonight is the light at the end of the tunnel. We've defined what is a great tasting tomato. We've defined how the plant makes those chemicals, and we've identified the genetics that underlie those chemicals. So we have all the pieces. Now we just have to do the long-term, the really long-term stuff, which is the breeding. Crossing them, waiting 4 months, making another cross, waiting another 4 months, putting all of the different pieces together into a package. But we know exactly what we're doing now. The path is there, it's all laid out. A couple of unexpected bonuses. This is the fun part of science. We had no idea that the brain talked this way to itself, that two different sensory systems could interact to influence what you actually think you're tasting. So the neuroscientists and the cognitive psychologists think this is great. And that's been the real fun part of working in an interdisciplinary project. We've released some varieties that really taste good. And we're getting, I don't have a Facebook account, but my tomato does. And it's great because people are sending us comments. They're sending us pictures of it's like looking at your kids. It's, sorry, Sarah. It's, people are sending pictures of these tomatoes saying, oh my God, I just harvested this bucket of your tomatoes and they taste great. And it's, that again, a different direction than I thought I was going as a basic scientist, but it's been a really fun experience. But I want to leave you with the thought at the very end, and that is we have made it our job to integrate the consumer into the breeding process. This is, believe it or not, revolutionary. We're doing it initially with all of the crops that the University of Florida breeders control: blueberries, strawberries, tomatoes. We're doing it with peaches. The things where we have genetics to back this up, we are doing it. But the process will lead to better products. And the sooner it's a long-term investment, but the sooner you start asking people what they really want, the better off they will all be. So that's been really exciting. Let me just say that this work has been funded almost exclusively by the National Science Foundation. I owe my life to NSF. They've really, we have couched this as a basic science project, learning a lot about metabolism, genetics, biochemistry, and they've been extremely supportive. And I have to acknowledge my colleagues, Linda Bartoshuk and David Clark at the University of Florida, and Denise Tiemann, who's been with me all this time, doing actually all the hard work, managing the field trials and all of that. And I do have a couple of USDA collaborators that I also want to acknowledge: Jim Giovannoni at the Boyce Thompson Institute, and Liz Baldwin, who's with the USDA in Florida. So let me leave you with this. This is the culmination of everything that we're doing. We've done two of them so far. We put on a dinner for the donors, the benefactors of the university. all made with things developed by University of Florida scientists, University of Florida varieties, and we bring in 3 top-end chefs each year, and we put on a dinner, and they feature all of our products, and it's just fabulous. This is the future of flavor. Okay, I need to stop. Yes.
The role of texture,
Uh-huh.
especially as you move up into the larger sized tomatoes. How important is that? Are you selecting for it, etc.?
Yes, that's a great question. I should have mentioned that. So it turns out that some of those outliers, like the Marmande that I was trashing, so it turns out that we have been able to come up with predictive models. So we can take the chemical composition of a tomato and predict how much people will like it. When we did that with Marmande, it was a classic outlier. The chemistry said it should have been a great tomato, and people hate it. It turns out it's texture. So texture can be a killer. If a tomato is mealy, if it's overly soft, it just turns people off. And so we actually do now incorporate. It took us a little while. We were a little slow picking up on it. But we now examine the texture in all of the varieties that we put through our consumer panels, and it's now factored into our liking. We actually asked people about texture, and that's actually how we got the first clue. They would tell us that the texture was just horrible on this one tomato. So the bottom line, that the texture can be a huge problem. In 80% of the tomatoes, it's not a problem. In 20%, it's a huge problem.
Early in your presentation, you were saying that the consumer wants cheap tomatoes year-round, but
Yes.
they're not willing to pay for it. But it seems that the beer industry and the coffee industry have discovered that people are more than willing to pay extra for the flavorful beers and flavorful
Yes.
coffees. So my question is, how do we know that the consumer is not willing to pay more for tomatoes? From what I've seen, we haven't really been given the choice.
Yes. So no, it's a great question. So the beer is a great example. I love to talk about craft beers. So craft beers are an example. When I lived in St. Louis, if you wanted beer, you had Bud Light, Michelob Light, or Busch. And I brewed my own beer because I wasn't going to deal with that. And now you go to the supermarket. Actually, when we bought our vacation home, the first thing I did was go to the supermarket and go to the beer aisle and say, what do they got? And that's a great example. It's also a great example because 80% of the beer that's sold is still Bud Light, Miller. But that craft beer market is growing crazy. I mean, the big companies are now buying up these craft brewers because of it. And I think that we should be doing that with tomatoes. But we've got to do it, we still have to do it cheaply. So probably many of the people in this audience are willing to pay more. You wouldn't probably be here if you weren't willing to pay more. But what we need to be able to do to really get where I want to be is I need to be able to give a great tasting tomato to the single mother who's on food stamps, who maybe can't afford to pay $5 a pound at Whole Foods. And so that's really our target, is the cheap stuff, making it taste really good. But again, you are seeing, so our tomatoes now, we're getting a lot of interest from small farmers who are supplying locally to restaurants, to farmers markets. Those people are starting to pick up on our varieties. So again, the craft beer analogy is a really good one. I think we're making progress. You've got to ask for it or they're not going to stock it.
One of your early slides, you mentioned the inverse of yield versus nutritional value.
Yes.
You've spoken mostly so far about flavor. Have you also been focusing on nutritional value?
We've looked at nutrition in tomato a little bit. So the answer is yes. We are screening, actually with my collaborator, Jim Giovannoni, we have an NSF grant where I focus on the flavor chemistry, he focuses on folic acid, ascorbic acid. We're working on carotenoids because they're the precursors of vitamin A. Actually, tomato is the #1 source of vitamin A in the American diet. It's because you eat so much tomato products. It's not that they're particularly rich in vitamin A, but we eat so many tomato products. So yes, we're really interested in that. The nutrient problem is a big problem in grains, for example. There's some nice studies in wheat that show that micronutrients are down in modern varieties of wheat. And so we are paying attention to that. I wish a lot of other people would too.
Some of these fruits that you're working on continue to develop flavor after they are picked.
Yes.
Others do not. Can you tell us a little bit about how that affects your scientific work?
Yeah, it's a very important factor. So I didn't mention one of the biggest problems that most of you probably already know about with commercial tomatoes. The field-grown tomatoes, particularly in Florida, they're all picked green. And it's done primarily because that's the best way that they can ship them. If you look at what's happened with the Florida tomato industry, the amount of tomatoes that are going to consumers has dropped precipitously because people are rejecting them. Because if you pick a tomato green, it does not develop full flavor. There definitely are genetic differences in the ability of that fruit to ripen off the vine. We've tried to minimize that because we can only work on so many things at one time. So we pick them specifically at just fully red ripe, and that's the way we do our tests. But you are absolutely correct. Some varieties ripen better than others. We've seen that in the commercial tomatoes. Some of the ones picked green can develop some degree of flavor, while some of them just are absolutely tasteless. So it's definitely a genetic component, but we have not been able to address that.
Thanks for a great presentation. I understand that ethylene oxide, which is used to take care of contaminated things and as an antibacterial in medical circumstances, is used to make the tomatoes red and ripen earlier. And I wondered, first of all, how that affects flavor, and secondly, whether or not lycopenes, which putatively have anti-cancer effects, are also part of the flavor process.
Yes. So first of all, ethylene is ethylene gas itself, not ethylene oxide, but ethylene itself is a natural plant hormone. It's produced, it is the ripening hormone in tomatoes. About half of the fruits that you would be familiar with ripen in response to ethylene. They're called climacteric fruits. Bananas, apples, tomatoes. The industry uses ethylene extensively. So they take those green tomatoes that they're picking in the field, they bring them to big warehouses that are the size of this room, and they pump ethylene gas in there. It's worse than it sounds worse than it is. It's a natural plant hormone. They do produce vast quantities of ethylene if they're permitted to. The reality is actually those green tomatoes don't produce as much ethylene as if they were on the vine. So they don't ripen as fast, and that is a factor. It's probably one of the factors that makes those green tomatoes not taste as good as a fully red ripe. We know from our experiments that there's more ethylene per fruit in something that's attached to the vine than anything you can add from the externally. Now, as far as lycopene, so lycopene's a great example. So it turns out that lycopene, let's keep there. It seems like if I stand in one place, it suddenly goes dead. Lycopene is the precursor of a couple of really important flavor volatiles. And those volatiles, it turns out, are ones that enhance the sweetness. So lycopene is really important to us. In fact, we are breeding now for varieties that have much higher levels of lycopene. We don't want to make health claims. I don't like to get into that area. But, well, you can make a claim that the tomato has significantly higher levels of lycopene and then let the consumer draw their own conclusions. But the fact is, we think that the ones with lycopene, higher lycopene, probably taste better. Again, that link to what's good for us and what we like.
I wanted to ask about the color of a variety. You know, for example, I grow purple Cherokees. At least to my perception, they taste better than a lot of other things, and they're darker. Can you talk about consumer choices and what people like and the colors? Thanks.
Yeah, that's a really interesting question too. So if you talk to the people in the supermarkets, the people who have to, to get back to your question about why don't we see this stuff like craft beer, the supermarkets only have the ability to put so many items on a shelf. They will only put so many different tomatoes on a shelf. So I have talked to some of the supermarket people and I say, well, would you like purple tomatoes? Would you like green or yellow tomatoes? They say, yeah, they look really good, but 95% of the people buy red, and the redder the better. So yes, they look really cool, and I suspect in some cases they do taste better because of what we do know from the genetics, from having the genome sequences, is a lot of those purple tomatoes, a lot of those brown tomatoes are very genetically related. They're someone, for example, the black ones or brown ones, all come from Eastern Europe. And so they share much of the same genetic pool. So if you like one, you may like a lot of them. But they look great. The chefs in the restaurants love them. But the reality is 95% of the people say they want a bright red tomato, and the redder the better.
Thank you for a great presentation. I'm really interested that you have a psychologist on your team, that you think a lot about those issues, and you showed us this vertical graph of preference with kind of worst taste at the bottom and best at the top, and the Marmande was down at the bottom, and you made some amusing comments about the French and their beliefs about their own superiority with respect to their preferences for food. And I'm just curious, when you said that, I think if I understood correctly, that if you could produce a tomato that averaged the preferences of your 100-person panel, that you'd be able to produce a tomato that tasted good to all of them, which suggested to me that there was not that much of a variation in the preferences within that 100-person panel. I'm curious whether you've explored cross-cultural differences in preference, and for instance, whether if you did the same test in France, whether you get different results, and whether the responses around texture as well or other factors might be different across those boundaries, and how you would see issues like that within the context of your research.
Yes. I mean, that's something we're really interested in, and we actually, if you want to watch a really, really interesting TED Talk, there's one about a guy, it's actually by Malcolm Gladwell. It's by a guy named, it's about a guy named Howard Moskowitz. And we work with Howard. And his whole thing is he gets in people's heads and tries to figure out what they want. And it's been really fascinating. So the best thing I can tell you to answer your question directly, we have looked at one set of ethnic preferences. We look at boring, white, Anglo-Saxon, Northeastern Americans, and we compare them to Hispanics. And it turns out the two populations like very different tomatoes. It's cultural. It's because what people in this part of the country who are Anglo-Saxon in history use their tomatoes for is put them in salads, put them on sandwiches. Hispanics use them in salsa. So we find that Hispanics like a tomato that has more acid, that's more firm because it stands up, because you chop it up and put it into a salsa. The people in this part of the country would have a preference for a big slicing tomato that they can cut up and put on top of a hamburger. So yes, we don't have a large enough population that we can really take apart a lot of the ethnic differences, but we have a hint that there are some very good ethnic foundations for liking different things. That said, I still believe what I said was true, that average of everybody is going to give you a pretty good tomato. It won't be necessarily your very favorite, but ultimately where we would love to be is to be able to get that down to market segments. We'd love to be able, I always draw, you walk down the soda aisle in the supermarket, or you go to one of those restaurants now that has the soda dispensers. You've got Coke, Diet Coke with cherry, Diet Coke with lime. That's the future. And that's what Howard Moskowitz is famous for. He actually worked to develop Prego spaghetti sauces. He put the chunky into the spaghetti sauce before we knew that we wanted it. And so that's where we want to be. That is the future, is to understand what you want and to be able to deliver that to you. That said, I still really believe, and I think the Garden Gem so far has proven it true, that when we get a very, very good tomato, everybody thinks it's very good. Yeah.
Hi, I was just curious if you could speak to soil quality and if you ever looked at that as a part of your research as well, and how that would impact maybe what would go into the breeder's toolkit, or also like speaking on the topic of hydroponics.
Yeah, I will tell you that I like to bash the Marmande tomato. The fact is that it probably doesn't grow the same way in Florida as it does in France. And so, what we would call terroir is hugely important. I was on this call-in show once, and I got a call from this lady who said, well, I grew up in New Jersey and grew up with my Jersey tomatoes, and I've never, ever tasted a tomato that comes close since I left New Jersey. And the very next caller said, I grew up in Indiana, and I have never tasted a tomato that like the ones that I grew in Indiana growing up. There's a huge cultural thing, but there clearly is an environmental impact. We know that. We like to think that Florida is the worst possible place to grow a tomato. The soil is really awful. The disease pressure is really tremendous. If it does really well in Florida, it's going to do really well everywhere. But that said, there clearly are regional differences. And so if you look at, for example, the commercial tomato breeders in Florida, they actually will breed for 3 separate regions. So they develop separate varieties for 3 separate regions within the state of Florida. So yes, there are huge differences. The people who grow wine grapes have known this for 2,000 years. Where you grow it and how you grow it can hugely impact how it tastes.
Last 2 questions. Yeah. First of all, how do you get these special tomatoes? Are you passing out seed packets?
I have 3 or 4.
Oh, good.
That's all I brought with me.
Second question is, I love the Honeycrisp apple.
So do I. It's the only one I like.
I've lived in Minnesota a long time. I think the University of Minnesota was involved. Did they benefit from the same methodology that you use? No?
Apples are really a completely different thing.
It's a long time.
Because they're self-incompatible, the genetics is a mess. They're apple breeding. Well, we have an apple breeder in the audience, I think, who might swear to this, but apple breeding is really hard. But that said, you see, to me, the Honeycrisp is a great example where you give the consumer something that they really identify with, and it takes off. And I think we can do that with tomatoes, but it hasn't been done yet. The supermarket near us, they put up a big banner. I'm expecting it any day now. The new season Honeycrisps are in. They put a huge banner on the front of the store outside in the parking lot. And it draws people in. And I think the Honeycrisp is fabulous. And I don't even like apples. And that's a great example where the consumer has driven technology.
Yeah, I'm sorry, we have time for just one more question.
I hope this is not an unpopular question, and it has to do with genetically modified
I'm surprised this is the first one.
products, and I thought I heard you say that you refuse to work with the part of the industry that grows genetically modified. Now, I want to say I don't buy and I'm not in favor of genetically modified fruits and vegetables. But I wanted to get a better sense of why you've refused to work with them because, is it sort of ethical reasons?
No.
Because, would that not be a way to get some of your discoveries out into the market more quickly?
Yeah, that's a very complex question. So the reason I left Monsanto was more a personal decision. It had to do with what I wanted to work on. I was involved in developing some of those crops that everybody's complaining about now. And I'm very proud of having done that.
Of working on them or of people
Developing them, making
disliking them?
No, making them. No, I was involved in making the herbicide, the Roundup-resistant soybeans and canola, and I am very comfortable standing up here and saying that I think that genetically modified crops have made more of an impact on agriculture than the Green Revolution. I'm not going to say that. I think many of the problems that people raise have more to do with industrial agriculture than they do necessarily GMOs. I think there are questions and issues that we need to address about how those products are used, and I'm not going to say that it's all rosy. I think that we need to be able to manage resistance to insect resistance, weed resistance. It's a very complex issue, but I will say unequivocally that in the entire 2 decades of GMO foods, we've been eating, all of us, unless you live in Mars, you have been eating GMOs for 2 decades. Over 2 billion acres of GMOs have been grown in the world, and not one single case of anyone in the entire planet ever having an adverse effect to eating those GMOs.
Why won't you work with them then?
Because the problem is one of registration. And so the registration process, so for Monsanto to register a new product probably would cost them a minimum of $25 to $30 million in getting approval from the USDA, the FDA, and the EPA if it's needed. Academics can't do that. We've been priced out of it. That's the reason.
So it's a money thing.
It is a money thing, largely a money thing that we don't want to go. It is also a little too controversial. So you see this, for example, do you know about the Arctic apple?
No.
So the Arctic apple is an apple that was introduced, approved this year. It has a gene in it, well, it actually it's missing a gene that causes the fruit to brown when it's cut. So you cut it and it doesn't brown. The Washington's, so this was done by someone in the Okanagan Valley in Canada, in British Columbia. The Washington State apple industry is furious with this guy because they think that now all people are going to think that all apples are GMOs and they're going to stop buying apples. I don't want to go there with my tomatoes. I'm copping out, and I feel a little bit guilty having let the genie out of the box myself. But I can't. I want people to adopt these things. I want you to eat this tomato, and if you tell me you don't want to eat a GMO, I want you to eat my tomato. That is a big reason why we're not using GMOs. That and the cost. So is it ethical? No. But it's wanting to get it into the most people's hands with the least controversy. I can't tell you, I've had people come up to me, sorry, I know we will need to break, but I've had people come up to me and say, oh my God, you're making hybrids. People don't know the difference between GMOs and hybrids. And this is an education problem. It's a serious education problem. I tell them, everything you're eating today was at some point a hybrid. Go back to wild corn and see if you want to eat that. But people are so confused, and the arena has gotten poisoned with misinformation. And I have just chosen, with our tomatoes, I just really want to get them in. I want to make a difference. I want people to eat better, and I want everybody to eat better. And I know that there is a population out there that won't touch them if they're GMOs. That's a big part of it.
Thank you, and thank you for your presentation.
You're welcome.
Well, that was wonderful. Thank you so much for a fascinating talk and question and answer session. Thanks everyone for coming. We have a wonderful plant biology department at Carnegie located on the Stanford campus in California. Take a look at our websites, our various websites, to learn a lot of other interesting plant science. And thanks for coming tonight, and safe driving to all of you.